Hello. In today's video, we shall study about bacteria. By the end of this video, you will have a clear understanding about features of bacteria, flagellation, spore formation, respiration in bacteria, reproduction in bacteria, and finally, economic importance of bacteria. Let us start with introduction. We all know that bacteria are microorganisms. It was Antony van Leeuwenhoek who first discovered bacteria with the help of a microscope. Louis Pasteur established the science of bacteriology. It is the branch of science that deals with the study of bacteria. Bacteria are omnipresent, that is, they occur almost everywhere in all habitats, water, air, soil, etc. We shall now look into the features of bacteria. Bacteria are minute, unicellular, microscopic, simple, most primitive prokaryotic organisms. Pro means primitive, karyon means nucleus, that is, the nucleus is not well developed. Each bacterial cell has a distinct, rigid, complex cell wall which provides shape and protection to the protoplasm. The cell wall is made up of proteins and carbohydrates. Chemically, it consists of acetyl glycoamine, acetyl muramic acid, and a peptide chain of 4 or 5 amino acids. Collectively, it is known as peptidoglycan. The cell wall may be surrounded by a distinct loose gelatinous sheath called slimy layer, or it may be a thick capsule. This layer protects the cell against desiccation and viral attack. Membrane-bounded cell organelles are absent, namely plastids, endoplasmic reticulum, etc. True nucleus is absent. There is a naked circular DNA known as nucleoid. This is the genetic material of the chromosomes of living cell. It is also known as bacterial chromosomes. Plasma membrane possesses respiratory enzymes and may give rise to infoldings called mesosomes. They help in the formation of cell wall, DNA replication, and also in respiration. You can clearly see the various parts of bacterial cell in the diagram below. Cytoplasm is present and contains many small gas vacuoles, stored food granules like glycogen and fats. Many bacteria have slender whip-like flagella originating from the cytoplasm. The flagella are used for their movement. Many bacteria also have accessory rings of DNA called plasmids. The plasmid DNA replicates and carry genes like fertility factor, that is F factor, nitrogen fixing genes, NIF genes, etc. The plasmids, which temporarily get associated with nucleoid DNA, are known as episomes. Mode of nutrition may be heterotrophic, that is, it may be saprotrophic, parasitic, or symbiotic, or it may be autotrophic, that is, either photoautotrophic or chemoautotrophic. They possess pigments like bacteriochlorophyll, carotenoids, etc. Sexual reproduction is by genetic recombination, that is, by conjugation, transformation, and transduction. I shall deal with each one of them separately. By the way, if you are getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, we shall look into the shapes of bacteria. Bacteria are of various shapes. Coccus, bacteria which are spherical, ellipsoidal, or oval in shape. They may be seen singly, Micrococcus, in pairs, Diplococcus, in chains, Streptococcus, or in sheets, Staphylococcus. Next is Bacillus. Bacteria are rod-shaped or cylindrical. May occur singly, Bacillus, in pairs, Diplobacillus, or in chains, Streptobacillus. Spirillum. Bacteria are spiral or coiled, bare flagella. Vibrio. Body slightly twisted like a comma. Flagella always present. Next, we shall take up flagellation. The number and mode of attachment of flagella varies in bacteria. The different types of flagella are atrichous, flagella absent, that is, bacteria does not possess flagella. Example, Lactobacillus. Monotrichous. Mono means single. Single flagellum at one end is seen. Example, Vibrio cholerae. Cephalotrichous. Many flagella attached at one end. Example, Pseudomonas fluorescens. Amphitrichous. As the name suggests, amphi means both, that is, single flagellum attached at both the ends. Example, Nitrosomonas. Trophotrichous. A group of flagella seen at both the ends of bacteria. Example, Spirillum volutans. Peritrichous. As shown in the diagram, flagella is seen all over the body. Example, Salmonella typhi. Next is spore formation. During unfavorable conditions, some bacteria like tetanus bacteria and anthrax bacteria produce resting spores known as endospores. They have protective covering. Botulism, a type of food poisoning, is caused by the germination of endospores of Clostridium botulism inside the food. During favorable condition, the endospore absorbs water, swells, and increases in size. The spore wall breaks, releasing a new vegetative cell. This cell develops into a bacterial cell. Next, we shall look into bacterial reproduction. First, asexual reproduction. It is by binary fission in bacteria. The cytoplasm and nucleoid divide equally into two. A constriction appears around the middle of the cell wall, resulting into two parts. These parts grow and form mature bacterial cells. Next is sexual reproduction, otherwise known as genetic recombination. A portion of genetic material, that is DNA, is transferred from a donor cell, which is considered as the male cell, to a recipient cell, which is considered as the female cell. Genetic recombination can take place in three ways: conjugation, transformation, and transduction. Conjugation was first described by Lederberg and Tatum in 1946 in E. coli. During conjugation, a part of the nuclear material of one strain is injected into the other strain. Former strain is called the donor of genes and the latter as recipient of genes. The donor or male possess sex factor or fertility factor, F factor. It replicates during cell division and is passed on to the next generation. It determines the formation of sex pili in donor cells and formation of conjugation bridge between the donor and recipient cells. If F factor remains free in the cytoplasm, then the bacterial cell is called F plus strain donor male, and if attached to bacterial chromosome, the cell is called HFR, high frequency of recombination strain donor super male. During conjugation, the two strains of bacterial cell come close to each other. The F positive male cell gets attached to F negative female cell with the help of sex pilus, forming a conjugation bridge. The F factor divides into two, one of which enters the F negative cell. Thus, the resultant is always F positive progeny. Let's revise with the help of the diagram. There are two strains of bacteria, F positive and F negative. When they come close to each other, the F positive strain, which is considered as male, forms sex pilus and a conjugation bridge. In the meantime, the F factor or fertility factor divides into two. One enters the F negative cell. So, the resultant is always F positive progeny. During conjugation between HFR donor and F negative recipient, the process in the beginning is more or less similar to the previous one, that is, development of sex pilus followed by formation of conjugation bridge. The chromosome of HFR breaks and both the strands begin to replicate. The daughter DNA enters the F negative cell through the conjugation bridge. If complete transfer of DNA doesn't occur, then the resulting zygote will not be converted into F positive strain. Next is transformation and transduction. Transformation was discovered by Griffith in 1927 in Diplococcus bacteria. A small amount of DNA from one bacterial cell enters into another bacterial cell and becomes integrated with its DNA. Transduction was demonstrated by Zinder and Lederberg in 1952 in Salmonella bacteria. A small portion of genetic material DNA of one bacterial cell is transferred to another bacterial cell through bacteriophage. A bacteriophage is a virus that infects a bacteria. Now we shall discuss about aerobic and anaerobic bacteria. Different types are obligate aerobes, can survive only in the presence of oxygen. That is, they need oxygen in the environment in order to survive. Example, Bacillus subtilis. Facultative anaerobes. They are aerobic bacteria but can survive in the absence of oxygen. That is, they are capable of switching to fermentation if oxygen is absent. Example, Pseudomonas species. Obligate anaerobes can survive only in the absence of oxygen. Example, Clostridium botulinum. Facultative aerobes, bacteria which can also survive in the presence of oxygen. Example, Chlorobium limicola. Next, we shall look into the different types of bacteria. Based on their mode of nutrition, bacteria can be broadly classified into autotrophic bacteria and heterotrophic bacteria. Autotrophic bacteria can synthesize their own food from inorganic substances. They can be further divided into photoautotrophic bacteria and chemoautotrophic bacteria. Heterotrophic bacteria cannot synthesize their own organic food. They are of three types: parasitic bacteria, saprotrophic bacteria, and symbiotic bacteria. Next is photoautotrophic bacteria. They have pigments and perform photosynthesis with the help of light energy. Example, green sulfur bacteria, Chlorobium species, contain pigment bacterioviridin. These bacteria, in the presence of sunlight, produce chemical sulfur by removing hydrogen from water. Next example is purple sulfur bacteria, Thiospirillum species, contain pigments bacteriochlorophyll and carotenoids. Next is chemoautotrophic bacteria. They manufacture organic matter from inorganic raw materials. Energy liberated is trapped in ATP molecules and is used to synthesize organic matter. Example, nitrifying bacteria. These bacteria get energy by oxidizing ammonia into nitrate. Nitrosomonas and Nitrococcus obtain their energy by oxidizing ammonia to nitrite. The bacteria Nitrobacter oxidize nitrite to nitrate. Other examples of chemoautotrophic bacteria are sulfur bacteria, iron bacteria, methane bacteria, etc. Among heterotrophic bacteria, first we shall learn about parasitic bacteria. They obtain their food from living cells of plants and animals. They may be non-pathogenic or pathogenic. Pathogens are microorganisms that cause diseases. Examples of some common human diseases are pneumonia caused by Streptococcus pneumoniae, typhoid caused by Salmonella typhi, tetanus caused by Clostridium tetani. Next is saprotrophic bacteria. These bacteria obtain their nourishment from dead organic matter. They break down the complex organic matter into simple soluble forms with the help of several digestive enzymes. Anaerobic breakdown of carbohydrates is known as fermentation. Breakdown of proteins is known as putrefaction. Next is symbiotic bacteria. Symbiosis is the association of two organisms in such a way that both are mutually benefited. Example, Rhizobium bacteria is found in the root nodules of leguminous plants. They reduce atmospheric nitrogen into ammonia, which is then taken up by the plants. In return, the plant provides nutrients and gives shelter to the bacteria. We come to economic importance of bacteria. First, beneficial effects of bacteria. Decay of organic substances. The bacteria help in disposing of the dead bodies and also increase the fertility of the soil. Second is, bacteria help in the conversion of atmospheric nitrogen to usable forms like nitrates, nitrites, and ammonia. Processes like ammonification, nitrification, and denitrification is done by bacteria. Free-living soil bacteria, Azotobacter, can convert atmospheric nitrogen into ammonia. I have already discussed earlier about nitrification, citing different examples. Denitrification, the nitrate and ammonia are converted into nitrous oxide and finally into nitrogen gas by denitrifying bacteria like Theobacillus denitrificans. Bacteria improves soil fertility. Decomposition of organic matter by bacteria help in the formation of humus, which improves soil aeration and water holding capacity. Role in industry. Many bacteria help in the production of lactic acid, curd, cheese, vinegar, etc. Medicinal uses. Bacteria are used in the preparation of serums, vaccines, vitamins, enzymes, antibiotics, etc. Harmful effects of bacteria. Food poisoning, botulism. Certain harmful toxins released by Clostridium botulinum causes symptoms like vomiting, paralysis, and death. Spoilage of food, souring of milk caused by lactobacillus, salmonellosis in poultry and egg are some examples. Pollution of water. Many pathogenic bacteria pollute water and cause diseases like cholera, typhoid, jaundice, etc. So today we have learnt about bacteria in detail. I have some practice questions for you. Please share your answers in the comments section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.